Okay, you're welcome, everybody. I welcome you from wherever you are, north, east, south, west of the continent. You're very most welcome. And before I introduce to you today's speaker, maybe let me tell you just a brief background of the country that she comes from. You never know, with the way phages, take us in all parts of the world, who knows, you might be in Uganda, and maybe these are some of the things that you will come and enjoy. So Uganda is a small country. It is found in East Africa. Sorry, I need to share my screen. Yeah. So you can see my screen, right? Can you see my screen, Emma?
Just press the share presentation view.
We can see. You can see it, right?
Yeah, we can see it, but it's the PowerPoint view. If you want to click at the bottom right, the slideshow view, that would be better.
Okay, okay.
Yep, perfect.
So that's perfect, right?
Great, yeah, you're good.
Okay, so you're most welcome to Uganda, the nation from which our speaker will come from. So it's a small country that is located in East Africa. And it is a country that is a hub of so much in terms of nature and wild and most probably phages. It is in the north, it is bordered by South Sudan, east is Kenya, south is Tanzania, and also west is Democratic Republic of Congo, and Rwanda in the south. So you will find 4 ethnic groups when you come to Uganda. You have the Bantu, you have the Nilotics, Hamites, and the Nilo-Hamites. Because of that, it consists of very many tribes. And as a result, there will be a diversity of a culture, of the culture in terms of the food, the dressing, the ceremonies. There's a diversity of this. The national flag, which has the 3 colors, black, blue, and black, red, and yellow, definitely do represent something that unifies the Ugandans as well as the Africans. And the crested crane in the middle is the national flag of Uganda. So the next time you come to Uganda and you're yearning to see some wildlife, please do visit us. You'll be able to see diverse wildlife. And those who really yearn to look at the big 5, definitely Uganda is the next spot for you to come and venture into. Uganda is also a hub of so many birds. So you want to do some birding, Uganda becomes the perfect destination for checking out, birds. If that is one of the things that you would be interested in apart from phages. So it is something that you would come and have a look at in Uganda. We also do have diverse cultural activities, cultural dances, quite diverse. And please, when you do come, we have a cultural center. We will be happy to host you and be able to enjoy as much of the dancing culture that exists in Uganda. I mentioned that there are 54 tribes, so there are lots of dancing skill sets in Uganda. So please do so. The next time you pass by, whoever hosts you, I'll ask them to take you to the cultural center. Another thing that we pride ourselves is the attire. Again, because we are such a diverse country, the attire is also diverse. So please make sure that you get one for yourself. The men get dressed up in, the robes, and the women adorn themselves. These are attires that are usually worn, in ceremonies and tradition, especially celebrations, and marriage being one of them. There is the Chiganda attire, there is the Chinyankore attire. So again, we get to enjoy a diversity of attires. Again, apart from phages, when you ever come to Uganda, please, there are quite a number of spots a number of tourism activities to do. For those who enjoy hiking, something for you. For those who enjoy water rafting activities, for those who would just enjoy watching cattle or seeing fruits or farm fields, fruit farm fields. There you go, there's something for you. So please, you're most welcome to Uganda. And last but not least, well, the last one is the university from which our speaker comes from, and that is the Makerere University. And why I needed to talk about it, because it is really one of the oldest universities, and it is a hub for so much innovation. Phages are one of them, but also the electric car and the electric bus that is soon going to be used, that is going to be commonly used in our country, is the innovations originate from Makerere University. So with this short, small background of Uganda, I do hope that I've given you a feel of what it is like, and please don't bypass us. Do come and pay us a visit, and not just for phages, but also there's so many things that I have shown you. So next, the next thing on our agenda is now that I am going to take this opportunity to not welcome, but I'm going to give a brief introductory welcome message for Dr. Jesca, who is going to be our speaker today. So Dr. Jesca Nakavuma is a Ugandan scientist with a veterinarian and microbiologist background. She serves as a senior lecturer at the College of Veterinary Medicine, Animal Resources and Biosecurity at Makerere University, Uganda. Her journey started off with a Bachelor of Veterinary Medicine, Master in the same discipline with a specialty in microbiology, and a PhD in microbiology at Makerere University. She has served different positions through the ranks of academic program coordinator and all the way to senior lecturer. Besides, she has held positions including head of department at the Department of Veterinary Parasitology and Microbiology, which stands at the Faculty of Veterinary Medicine, Makerere University. She has also held the position of chair at the Department of Biomolecular Sciences and Biolab Sciences, again at the College of Veterinary Medicine, Animal Resources and Biosecurity at Makerere University. She has served the position of Deputy Principal, College of Veterinary Medicine, Animal Resources and Biosecurity, again at Makerere University. She prides herself in teaching for over 30 years in microbiology-related courses and has supervised over 80 students of which these include undergraduate, master's and PhD graduates. She has gone ahead to publish over 50 articles in peer-reviewed journals and participated in several conference proceedings, as well as authored in 3 book chapters. Her success in grant acquisition is commendable for an African scientist. She has won over 10 grants, with the highest fetching her over 900,000 USD by the and was granted by the European Union, African Union funders. And this was in the year between 2018 and 2021. So through her collaborations with the University of Ghana, she and her team have engaged in research that will develop phage cocktails as disease control applications. Because of her research expertise, she has gone ahead to form and lead the Phage Research Uganda team in Uganda. And because of that, she is at the forefront of, most of the activities that occur in that team. She continues to extend her services to several professional bodies, including but not limited to the Uganda National Technical Advisory Committee on Vaccines and Immunization, Mentoring and Empowerment Program for Young Women, the National Drug Authority in Uganda. She's a senior ambassador for Uganda for the International Board of Ambassadors of the International Society for Microbial Ecology. She's a member of the Phages for Global Health, a non-government organization that trains phage skill set in Africa. She serves on the editorial board of the journal called Phage, Therapy Applications and Research. She continues to serve to the community through the Rotary Club, conferences, workshops, farmers, university students, and phage researchers. Her research interests cover a range of microorganisms focusing on biocontrol and industrial applications. And not only does she enjoy research and teaching, she also enjoys a good hearty laugh with anyone around her. At least I testify upon that. So with that all said, please join me in welcoming today's speaker, Dr. Jesca, for today's webinar, where she will talk about the challenges and possible solutions in phage research, and most probably share her research experiences from an African context. You can have the floor, Dr. Jesca.
Thank you very much, Rita. Let me share my presentation. Is it there?
Yes, that looks great.
Yes,
Oh,
yes, we can see.
Yeah, I had to modify a title after getting some of the questions that colleagues would like me to clarify. So I just put it as phage research in a resource limited setting, practical challenges and possible solutions. The challenges and solutions may not be so much, but the experience is what I'm presenting more. So my presentation, I have the introduction and the phage research experience, what we've been involved in since we got enrolled into this area, the search for funds, the human resource and collaborations, challenges that we have encountered, and the transition from basic research to applications, and I will end by acknowledgements. So it was in 2017, June, when Dr. Betty Kutter came, I think visited Tanzania, but decided to move through the East African countries. So she came to Makerere. We had a summer school and a conference during that period. Being the deputy principal then, I welcomed her and offered her audience. We organized for her talk, and a number of people became captivated into what she presented. So, and that is the journey where our journey in phage research started. So when she left, it was mainly the undergraduate students. I don't know whether my cursor is being seen. The undergraduate students who showed a lot of enthusiasm, and we formed a team preliminarily, and we called it Phage Team Uganda, which still stands. And the aim was for us to carry out research and development to combat antimicrobial resistance, and to look into the issues of food safety using the phages, and targeting, and in this we're targeting the animal production systems because that is where most of us, our niche is. But we also have the laboratory students who are interested in the clinical settings. So the students identified some special projects for their final year research. And these projects depended on the programs that they offered. So these are the pioneer students. I think there was one missing. Paul Mutumba was a laboratory technology student. He looked at the Pseudomonas aeruginosa from surgical wounds. Just because one of the captivating topic that Betty presented was treatment of the wounds and the diabetic wound. So he went into that. Then Nelson Ibukamba was a BVM student. He looked at Salmonella from the Mount Gorilla ecosystem. He was so interested in the gorillas, and he thought they can transmit pathogens to humans as well as the humans transmitting pathogens to them. So he's currently working with the gorilla, the mountain gorilla projects. Then there was Gideon Muhindo, not Muhingo. Then he offered Bachelor of Animal Production Technology and Management. So he looked at the E. coli, Escherichia coli, and Salmonella from the swine farms and from the swine abattoirs. And then the only lady who joined the team was Immaculate Nakavuma, also a Bachelor of Animal Production and Technology student, who looked at Staphylococcus aureus from the dairy cattle with mastitis. So there was another vet student, Ismail Asimwe, who looked at enterohemorrhagic E. coli from the dairy cattle. So those were the first students who really were involved, but there were some who had just graduated who were in the lab and were equally involved, and they became converted to join to do research. So thereafter, in July 2017, the first East African Regional Phage Laboratory Training Workshop was held, and it was hosted at our college. I think many of us can recognize Tobi. This is Martha, who was a trainer, and Benjamin, and then Janet, who is a postdoc student of Martha. So we had participants from Tanzania, Kenya, Uganda, and one Ethiopian. Being the host, I took advantage and involved more participants compared to the other countries. After all, there were no expenditures on travel and even accommodation. So down here is the other participants when we were embarking on the laboratory work. So before the training, we had this uppermost figure may not be very clear. We had tried to isolate bacteriophages because Betty sent me some money to buy a few of the expendables. So we tried out, but the students were looking up to me, thinking that I knew, and but I was also as naive as they were. So we didn't know exactly what the phage plaques looked like. All I had in mind, there is a zone of inhibition. So it is this second from the left, the second column. This is where I was confident that those are bacteriophages. But the first one had a rim of clearance, and I was wondering. So we just took these photographs, although not very clear. I sent it to, there is a gentleman in Egypt who has done a lot, I've forgotten his name, who has done a lot on phages. He told me that the first one is likely to be maybe the bacteria producing bacteriocins. So, and we had some other plates that had smaller, much smaller plates like what is on the left on the lower one. And we didn't think those were actually phages. So those ones were just throwing away. And after the training, that's when or during training, that's when we appreciated how the plaques looked like. And we went ahead, as you can see down here, to be involved in more or less professional phage work. So thereafter, so that the first lot graduated, here we are celebrating a small cake with them. And we formed a journal club. It had somehow hibernated some time back. And just because after this first, the pioneer students had to look for a way of living, they had to get jobs. So we were like dispersed, and activities in as much as phage research was ongoing, but the phage team journal club did not continue. Then somewhere down the road, Dr. Tunga from the Primate Research Institute in Kenya came and visited for 2 weeks and was working on his phages. So he also offered a lecture to the undergraduate veterinary students. And these are the ones who are coming to me that they want to do phage work. And I ask them, how did you know about phages? And they remind me, you remember I brought us a visitor who told us about bacteriophages. Then courtesy of Betty Kutter, she organized for funds and invited the trainees, some of the trainees from the first East African training workshop to go to attend the 22nd Evergreen Biennial Phage meeting. I tell you, when we listened to the presentation, I was just amazed, finding that some people are looking at synthetic bacteriophages, and here we are still looking for the natural ones. And I was wondering whether we can get where they are, but with the collaborations, I'm sure we shall get there. So in 2018, in December, Betty came back again and visited. So these are some, this was on the first lot. This tall guy is currently doing his master's on the AU project. So this lady in between me and the first lady student, pioneer student, worked on E. coli from chicken. So, but we couldn't go far, we had no funds. However, her work gave us some baseline information that we used to apply for funds when the chance arose. And someone behind here, not very clear, is the one who, a master's student who took on to push the APF research further. So then we went into grant hunt, searching to hunt for phages. So after, I think when we were still at Evergreen, the team that was there, we thought of responding to a call that was from NORAD to look into the alternative methods to curb antibiotic resistance bacteria from farm to fork in East Africa. There was a short time to the deadline, so we did not really think through clearly or have time to think through and focus and have our proposal very clear, so we were not successful. Then there was another call, and it was the Students' Innovation Challenges. So I got one of the students who qualified to, and we prepared a proposal having the smart vibriolytic phage, where we thought that we can get bacteriophages that can be added to the latrines, because most of the cholera cases occur after heavy rains and washing away of the latrines, and thus contamination of the water. We were unsuccessful. We tried to resubmit it to grant challenges called by Bill Merinda, still were not successful. Then the third one is what I had already mentioned from the previous slide, the undergraduate student who worked on avian colibacillosis. When the competitive grants from the Makerere University Directorate of Research and Graduate Training pushed out a call, I applied and I got $5,000. So I enrolled a master of students who looked at the molecular bit of the molecular characterization of the E. coli, and this student will be graduating this May. Then after there was this IDRC Innovates AMR call, where we organized some universities from Kenya, Rwanda, Uganda, and we got a Canadian collaborator. So we wrote about probiotics and phages for use in aquaculture in East Africa in order to curb or reduce antibiotic use. and therefore curb antimicrobial resistance. Still were not successful. This was about $3 million. So I think we cannot write proposals worth that much money. Then we went on, afterwards there was a call from the EU. Indeed, there were, AU, the funders are the European Union. So the way the call was, we looked at it with some of my colleagues and we just gave up, because you write a concept and an application at the same time. So whereas normally it's a concept, and when you go through, you write the full proposal. So we just pushed it aside, that shouldn't waste our time. It requires a lot when we're not sure of going through. However, the Ghanaian team contacted me as a collaborator. So I joined them as a collaborator, and they had an idea of food safety, looking at fish spoilage and reducing those microbial counts in fish. And for me now, I brought in my veterinary perspective of production diseases. So we ended working on production diseases, and I think amongst themselves they met, and probably they realized that I better fitted to be a PI, and we had to push this proposal through. Fortunately, yeah, we were funded. So we have institutions from Ghana and Uganda involved, but we also have Martha as the UK advisor. So this is the team on the Safe Fish Project, the African Union Phages Against Fish Pathogens Project. We short named it Safe Fish. There is Evans. I will not murder the other surnames. I will pronounce them, and that will not be good. So I'm only using their first names. Evans is from Food Research Institute, Ghana. Then there is Otonyo from the Water Research Institute. Deborah is from the Food Research Institute. Isaac is from the University of Cape Coast. Susan is from the Noguchi Memorial Medical Research Institute. Then Martha Crocky from UK. So, and below is the Ugandan team, Jesca as the PI. We have Dr. Claire Mugasa, a molecular biologist. Maureen Mayanja is a social economist. And Samuel Ponsian did his PhD, is a vet. From Jesca to Samuel, we are all vets. We did undergraduate, the BVM course. Samuel did his PhD in fish pathogens, bacterial pathogens. There was one of the trainees. He wasn't a trainee, he was the technologist, but he grasped much more than even us, the one who were training, and he's very good at it. So he has just finalized his master's on microbacterial phages. And then Dr. Johnny Walakira is from the National Fisheries Research Institute. So that is the team, multi-institutional, multidisciplinary. A project of that magnitude needs such. So the aim of the project is to develop bacteriophage cocktails as fish disease biocontrol agents for improved aquaculture activity, productivity, not activity, among tilapia fish farmers. So, but our expected outcomes is for us to have awareness and buy-in, because we know phages is a new thing on the block, to identify the bacterial pathogens and their distribution within the countries, for those of economic importance, the drug use in aquaculture and the antimicrobial susceptibility. We also plan to have our local or in-house phage banks, and we expect to have 4 phage cocktail products formulated, 2 in Ghana and 2 in Uganda. But most importantly, also the research and development of phage technology capacity should be enhanced. So we, of course, the money given is not enough. It sounds big, but it isn't. So we've gone ahead wherever we see some calls, we respond to them and identify some areas that can fit within. And we work as a team down here, the Uganda team, Safe Fish team, to develop the proposals, identify who is best suited, basing on the core requirements. So we have the Makerere University Research and Innovation Fund, which is a government of Uganda fund. And in this case, we are looking at the in vitro lab evaluation of bacteriophages, their activities within the lab. And then here we selected only the Eromonas because of the amount of money offered. Then we have the Makerere Directorate of Research and Graduate Training, supporting early career academics. This is SECA. And the funds are from the Carnegie Corporation of New York. And in this case, we are evaluating the Eromonas phages in the aqua tanks. And the PI whom we brought on board is Dr. Andrew Tamale. So, and below here, the one with the black mask is his student. And the one with the maroon mask is he has been an undergraduate student who has worked on phages, and he's taking him through the steps of phage isolation before he starts work in the aqua tanks. So the experiences we had with or I have had with grant hunting. The first thing is to select the right collaborators. The institutions must be sound enough, and also you can have a mix, the upcoming and those that are well established, because that's where the funders can have confidence that these people will be able to deliver. So we look at all the experience or the skills of the individual members in order to fit or to have a sound team that can compete favorably. So I've also experienced that the more the amount of the research fund, the more requirements, and it needs intensive preparation. And in our case, we usually have limited institutional support, yet a lot of institutional information is requested. So it becomes this coupled with the fact that we usually respond late. So there isn't enough time. So it is a stressful situation when we are writing those grants. What I also realized was that we try to concentrate on the technical information, the science, and be very good. But then we leave out other issues like information management or the budget itself. So in the end, you find that there are certain things you did not take into consideration, which will affect your project implementation. Then the research team members, usually we have various priorities and deadlines. So when it comes to proposal drafting, you may not have all of them fully involved, despite the need for multidisciplinarity, multi-institutional composition, but one or two persons at the core of it can organize and put the proposal in place. And I also realized that when a call is on, that's when we get the energies right, or the vapor right, and think properly. And usually you may not be successful, but we do not lose hope. We just modify and submit elsewhere. The lessons learned in there were some lessons as well. So it's that you have to note the theme or study the theme of the grant. And even if you had a proposal on a shelf, you can easily modify it and write convincingly, get a grant. And when you are just starting into this grant writing, it's better to start with small calls or maybe the institutional-based ones are a bit easier and there is minimal, not much competition. And since phage research is still novel in our setting, we usually get funding. With these small grants, it helps to build the CV and anchor you into phage research, or to convince the funder that you have experience in working on bacteriophages. Is more especially if you have published an article. Then building networks. I think the regional trainings, these phage trainings helped us a lot. And also trainings in other areas, because I got to know that other colleagues and workshops organized by the International Committee of Food, Microbiology and Hygiene. Also at the conferences, even if you did not get a chance of talking to someone, you just get an abstract book and see if there's someone who has similar work as you are interested in. And you just write to that person at this conference together, and we presented on this. I'm interested in such work. And what I've noticed with the phage people, they are amazing, the phage researchers. They are so helpful, they are outgoing, they will really help. Then collaborating with universities is a plus because of the students. You offer students the research materials and they help you a lot in implementation when carrying out research or for your project, the research activities for your projects. So in form of capacity building or where we've reached, I've mentioned Deus Kamia, the lab technologist. He will be graduating this May with an MS in immunology and clinical microbiology. He's the one who looked at the phages against M3B biofilms. Then George Kaziwe did, at his undergraduate, he did pharmacy. So then he joined the molecular biology and biotechnology master's program. And he's the one who worked on the APEC, and he got so much interested. He's trying to see how he can infuse his pharmacy knowledge in us having therapeutic products made out of the phages. So we have other students who are currently undertaking the study again attached to our projects. I've gotten others from other institutions and programs who are requesting for my supervision to be on the supervisory team. And I'm on a doctoral committee for one of the PhD students who is looking at phages against E. coli and Klebsiella pneumoniae that are against the carbapenem drug. So for the linkages, Betty Kutter, Tobi, Martha, Evaliensen, Sen is not there. Then Jesca Nakavuma. These have been really helpful. I often write to one of them if I need someone to help with in certain areas, and they forward particular emails, and you try to contact those people. At times it's not successful, but in other cases it's successful. Like for the APEC phages, we had whole genome sequencing. Intellectual Ventures in Seattle helped us, but we've also contacted some phage manufacturing companies. Just because we've not gone far, we've not engaged them fully to see how they can help us with the product development as we put our house in order. So you can also contact authors. Those articles you commonly read, there are some salient personalities that are writing that have been anchored in phage research who can be of use. And then there are some phage groups that exist, like when we were in the US, I saw those that are working on the cholera bacteriophages. They had their meeting, mycobacteriophages. So if the pathogen you are working on, there is a team, it's better you get in touch and start collaborating or getting advice from those. So, and I think it is this mycobacteriophage, I mean, not the bacteriophage research, and also being on some of the committees that have introduced me to people, because that's how they got to know me. I'm on the editorial board for the Phage journal, then for the International Journal of Food Contamination. And yesterday I received an appointment letter from our Vice Chancellor to be on the committee to develop the research agenda for Makerere University for the years 2021-26. And I feel probably, for me, I have a feeling it is phages that has pushed me far. Then we have the various journals where we can publish, depending on what. If it is entirely phage, you might find that you do not fit in that particular journal scope, and but others are quite broad. They have a broad scope that you can fit in. So it's the Phage by Mary Ann Libert Publishers that is dedicated. I think it's probably only the only one that is dedicated to phage research. The only one I'm aware of, let me put that. And this is it's a top cover. It might change depending on the issue, but these days it comes with this. So which can, and we can easily publish in there. We had a meeting last week, and I was informed that it is still free until the end of next year. So I urge my colleagues to take advantage. Let us fasten whatever we are doing, and we publish before that time. So the year ends. So we have some general challenges. Some are institutional. The infrastructure, we don't have a lab space dedicated for phage work. So we work within what is available, very often congested. You have the equipment you are sharing, and you find you need an incubator and it is already full. Certain equipment that you may want are not available, a shaking incubator, and yet the funds you have may not suffice to acquire all that. And that can also affect our upscaling. Do we have the facilities for upscaling? Then the other one is the human resource. Some of these projects, especially the small one, you may not have room to hire persons. Yet you need someone who has some knowledge on phage work and who is probably continuously there to supervise the students that you've enrolled. So, and that becomes a problem. And also that lack of funds. It's the same, probably the same reason as to why I lost the pioneer students. They would have joined, like if I had an organized program with the funds, they would have continued with the lab work, because up to now they are still interested in bacteriophages. So the training support, you may get funds without scholarships, and yet the students need to pay tuition, they cannot afford. So you miss out on very good students because you cannot, but you cannot support them in that area. But what I usually do is to advise them that you apply, get an admission letter, a graduate program, and use that one to look for funding, and many of them have succeeded that way. Then the other challenge are the relevant policies. I don't know whether in some of which African country has maybe a policy that is now catering for phage use. I understand in Kenya they had embarked on it. We are still wondering, even if we get a product, we may have a hindrance as by the policies or regulatory frameworks for phage use. And we are also wondering, should we fit within the Uganda National Bureau of Standards? Should we fit in a National Drug Authority? Or because there is a ministry that is Ministry of Science, Technology and Innovation. But they are looking more of science and innovation, but the advocacy and the regulatory frameworks for some of these emerging innovations do not exist. Then the other one is awareness versus acceptability. Once someone hears a bacteriophage is a virus, then you've killed it all. Someone told me, for me, I cannot eat your food, even if it is safe, if you have added bacteriophages. So we still need to have a lot of advocacy amongst our people to create this awareness and acceptability. Then transiting from basic research to applications. So we are talking about cocktail formulations, but evaluating or characterizing these phages is still a challenge to us. No transmission electron microscope amidst us. So one of the requirements or the recommendations is to have phages in the cocktail having different attachment sites. So I can come up with my formulation based on the combined activity as far as cost range is concerned, but are they attacking different sites? If resistance comes in, would they be knocked out or at a go? So all those, and also the whole genome sequencing, we are still, it's not very expensive, but still some without funds you may not afford, and yet you cannot go further with a bacteriophage if you are not sure of its safety. Then we have the other challenge that we are thinking of, this bulk production of the bacteriophages, stabilization, appropriate packaging. So all those are still far-fetched as far as we are concerned, but I feel with the collaborations that this can be handled. And then the last one is acceptability, the cost-benefit analysis, studies, environment assessment. Definitely when you produce the product, they will need a report. In as much as we say they are naturally available, they are very specific and so on, still the National Environment Management Authority will require a report before we produce. The marketing, the intellectual property issues, and getting industries to collaborate with. Many of our industries, if we want to produce bacteriophages as antimicrobial agents, they are in place conventional drugs, and they are just compounding and packaging, not that they are producing or manufacturing as such. So will we be able to penetrate them with our bacteriophages? And so all that, we still have a long way to tackle those particular issues. So I acknowledge or I thank the administrators of Africa Phage Forum who have given me this opportunity. The Betty, Tobi, Martha, who grounded us, or the team into phage research. Phage Team Uganda for having the trust in me to steer our activities. The Safe Fish Research Team, where we are, which is I think the biggest we are handling as phage researchers in our country. We've received various funds from various organizations. I acknowledge all of them. And the participants from this continent and beyond, I thank you very much for your attention. That is the end of my presentation.
Thank you very much, Jessica. I don't know if you can hear me.
Yes, I do.
Yes, so this is Angela Makumi, but apparently because I have a blackout, well, my video will be very limited. However, I have looked into the participants and I have looked into the questions, and I think if you would like, I can involve the participants who can maybe clarify on especially when it comes to cocktails, because I've seen Rob Lavigne is present and I have seen Tobi is also present.
Oh, okay, that is fine.
Yeah, so we can engage them in asking a few of the questions because we are very limited on our research, but maybe they have options they could give us on what we could do better, because everybody like on the, I would just, because we're limited of time, and I think there are more of the experts in, because they've done a lot of phage sequencing and cocktails, I think they would be more enlightened to give us ideas on how a resource, like we are very limited on resources, and how we can help ourselves in terms of when it comes to cocktails, especially when we cannot do sequencing and we cannot do TEMs. So if that's okay with you.
Yeah, it's very fine.
Yes, maybe they can share different opinions on, so I have a few questions, and wait, I'm just looking at the questions that I had were on phage cocktails, and maybe I can, if Rob Lavigne is available. I don't know, Rob, because I saw he was one of the participants.
Just a second.
I don't know if he'll be available.
I need to make anyone who you want to talk a co-host. So just let me know the names. I'll make Rob a co-host if he wants to.
Yeah, so I would like to talk to, because a few of these questions that are raised on the chat box, maybe Tobi might be able to explain more, because she did part of the training. And Rob might be able to explain more.
I'm here.
Ah, thank you, Rob. So apparently, because we are resource compromised, not really compromised, but because we don't have the resources, do you think a TEM is likely to tell you what phages need to be in the cocktail? And also, if we can't, like, what are the best in a resource strained country? What is the best way? Because I know that it's always a strong fold when you need, when you have a European or American partner involved. But imagine if this is Africa and we're trying to create our own research, but because we have a resource restraint, is there any other way except TEMs and sequencing that we can maybe say this ones might fit into a cocktail?
Okay, thank you. First, my appreciation for the nice and comprehensive presentation. So it's good to see that all facets are being covered and thought about. I personally think TEM is not needed anymore. By contrast, the capacity to do sequencing is not only suitable for phages, but also for bacteria. I think here one solution may be the nanopore sequencing, having a handheld device which prevents the expensive cost of equipment might be an option. And still, it costs some money, but pooling phages allows you to sequence phages at around 15 to 20 euros roughly per phage. But still requires some, from the bioinformatics side, some specific knowledge and know-how. I think that's important. Secondly, one of the only things I didn't necessarily agree with is why automatically focus on small grants? They are taking in my experience, roughly the same amount of time to prepare compared to bigger grants. And yeah, I wouldn't be opposed to seeing more taking a chance and ambition there. And you raised the point yourself, Angela. I think partnerships with other groups and so forth can help there.
All right.
So get rid of them. It doesn't reveal much, and rather focus on the new low-cost sequencing techniques.
And so what if we cannot afford? like, is there another way that you could think about apart from the host range that could actually tell you that these are different, these are not, what we need to, because I know also putting a cocktail, you might be limited maybe to 7 phages, 8 phages because of what we would need to become a product, because it involves a lot of, and a lot of people tell us like you only can have 5 or 7 limitations.
Yeah.
Except when it's for plants and everything should be.
As a starting point, you could think about being very strategic in the choices of pathogens. For example, Staph aureus, if you're thinking along those lines, does have phages that cover 80 to 90% of isolates. So if you can develop applications there using only one phage, or focusing first on, let's say, low-hanging fruits, that can help. The same is true, that's medical, of course, and that's maybe more far off, but the same is true for aquaculture. The microbiological properties, being able to produce phages that have high titers and require less purification. And when we think about maybe aquaculture or things like that, where the need for an ultra pure product that would be needed for medical use, yeah, I think there are some microbiological parameters that you can select for, and the highest titers, the best producing ones, to cut costs in your cocktail design.
And talking about low-hanging fruits, do you think in Africa we should go for maybe topical livestock? And what is your opinion on human phage research? That's just talking about the low-hanging fruits.
Yeah, it depends on the need.
No, in terms of
it's difficult to assess in a general statement, but yeah.
No, but because of the research involved, I think with humans it would be having more implications. But if it's aquaculture or livestock or plants, then it's easier. If you have a product, it's easier to license. But I think we also want to go into human research. But what would you think are our disadvantages? Like, I know Europe already is trying to go through it, but you're already having limitations.
I can chime in here too.
Yeah.
Hi, this is Tobi.
Yes,
Nagel.
Tobi.
Can I chime in?
Yeah.
So another piece of it, if you're thinking about the final product, is the cost of manufacturing that product and the regulatory hurdles. And manufacturing a product for human application as opposed to, say, food decontamination, which might include poultry and crops, or other types of animals. Usually the purification for human applications is much more expensive, maybe even 10-fold more expensive, because it requires GMP manufacturing, whereas for food applications, it doesn't. And so I think that makes it just more tractable from a sustainable financial approach for a business. Also, there are already approved products for food decontamination in the US. And since many developing countries follow regulatory guidance from the US and Europe, they would be more likely to approve that, I think, food applications right now, rather than human applications. I can that the Kenya Ministry of Agriculture is very interested in applying phages in agriculture. So hopefully other agriculture leaders are as well. So I think I totally agree that human application is important, but in terms of getting this going in Africa, it might be easier to do it through non-direct human applications.
And with humans, do you think topical would like give a foot in? Because with topical applications, we might treat them as herbal or bio products because they are found in the environment, and then we can say this is a bio product. And then
Yeah,
we can
and-
treat them as either herbal or a bioproduct.
And it's the same, sort of the same answer as I just mentioned, in that topical products don't require as much purification typically for regulatory agencies as do products that are taken into the body, especially IV or otherwise. So again, I think that would be an easier first step.
All right, and Tobi, we have a very, someone said we just have to hit it where it hurts. I have a question for you. What is the actual challenge of phage research in Africa, and what is the way forward to get out of the wood? And a similar question is, when the first West Africa Phage workshop was held in Ghana, okay, there was a talk on safe fish. Phages were isolated during the workshop and were shipped for purification in the UK. So at what stage of purification is it since then?
Sure, and I'll start with the second question. I've sent a WhatsApp message to Martha and Janet Nale to ask them to join this if they can. Martha will join in a few minutes, I think. So they've been doing the purification and the characterization in their lab. They've also been doing that for the two previous workshops, the phages that were isolated at the Uganda workshop in 2017 and for the Kenya workshop in 2018. And I know that they are backlogged. For the last year, they've had limited time that they were allowed to work in the lab because of COVID. So I suspect that's the simple answer, that they're back. Oh, Janet's here now. Janet, you want to answer? Jessica, if you can make Janet a co-host.
Yep, I will, 1 second. Okay.
Oh, hello, everyone. Apologies, I didn't see Tobi's message earlier. So yeah, regarding the characterization, because of the COVID, basically we spent almost like 8 months away from the lab. So our own personal research actually are under some kind of restrictions at the moment. And even the lab occupancy, we only allow a certain number at a time. So we have to really prioritize work that we've been seeing. That greatly affected the characterization of the phages for the Kenyan workshop and even the subsequent ones. But for the Ugandan ones, when we did it, I think it was 2017, and we brought back the samples. So we were able to purify the samples furthermore, because during the workshop, we only were able to characterize, I think, up to 2 or 3 plaque purification steps. So I brought those samples back and then further purified them. And even to for the other samples, we lost the phages in there. So I have to go back to the samples for the group, the actual original sample from the sewage, to isolate phages for them. So in total, we're able to purify about 20 phages from that workshop. And then from there we did TEMs and we sequenced I think 5 of them. I got a personal grant to actually sequence the phages, and then we did some host range analysis as well for the phages. And actually for the Kenyan samples, I think we did we actually did some work. That was before the COVID lockdown. We did some work on the Kenyan phages. We purified up to, I think, 7 phages. And we even optimized a cocktail. These phages targeted Pseudomonas. So we optimized a cocktail that could eradicate an ATTC strain. And then one other strain that we have been using in the workshop, I think is WB1. So we've been doing some work on these isolated, but it's really difficult because of the COVID. So apologies for that. Thank you.
Thanks for getting that personal
Janet.
grant for that, Janet. And I understand that your other grants required you to finish other work before you could get to these new ones. But I think we should all be keeping our eyes open for additional smaller grants to help with the further characterization. So if any of the folks from West Africa have suggestions for collaborative grants from West Africa, we'd be happy to apply for those for the further characterization.
Tobi, another question for you is, when you're having the training sessions through Africa, through Asia, do you think you should add more like difficult bacteria to get phages from than tackling the normal E. coli, Salmonella?
Well, since our whole goal is to introduce people to phage isolation, and many people in our workshops have never done it before, I think it makes sense to start with the easiest ones first, and then they can move from there towards the more difficult ones. And our workshop instructors and various collaborators around the world are always willing to provide guidance remotely as people start working on the more complicated ones.
And another question is, this is just a general question. It might be for Jessica, Tobi, or Rob. Are there linkages that we can benefit from that have to do with the use of phages for diagnostics or vaccines?
Personally, I think the use of phages for diagnostics is a great potential, and maybe even pairing a diagnostic with a specific phage cocktail. That's my 2 cents. I wonder what the others think.
Jessica?
Yeah, thank you very much. There are a number of teams that are carrying out research developing phages for diagnosing, I think for monitoring water quality using the E. coli phages. So it's possible to develop those diagnostics, but you need to get to the right people who are involved to give you the details on how you can approach it. But phages have been used as diagnostics for some time, except now that they are modified much further.
Yeah, and I, for one, I also got a grant on using vectored vaccines, but vector means a phage-based vaccine in poultry, which is still ongoing. Yes. And Jessica, another question to you is, if you were going to use cholera phages in a pit latrine, and the fecal matter is very dense, so just as you had mentioned on a grant, the fecal matter would be very dense because you have accumulation of feces over so many times. So how would you propose that, because the phages cannot only be at the top, how would you propose, because like in African settings, everyone has a pit latrine. What would be the way forward to make sure that, Yeah,
there are some products that are on sale, and I guess they are enzymes. So the instructions is like you mix in maybe 5 liters of water, then you apply overnight. But like the phages are applied on a diabetic foot wound, and it penetrates wherever the pathogen is. I would assume it can also penetrate. Besides, it is not as solid because the microbes are busy breaking it down. So it's not that solid material as a rock that phages cannot manage to diffuse to some areas.
all right. I just, I've seen also so maybe this might be the last question because we are over time. Given that research development tailored to addressing human health have always attracted attention of everyone, including policymakers, pharmaceutical industries, etc., do you think more attention of using phages to human applications would be the easier ways to lobby for phage acceptability?
Again, I think this is Tobi. I think lobbying, yes, I think the public and the policymakers might understand it more if we talk about direct human applications. But there's also the realities and the costs and the technical challenges of drug development that are just more complicated for human applications.
Okay. Jessica, a final question to you is.
Can I follow up?
Yeah.
Yes, Tobi, based on what you're saying, is it not difficult? Are you discouraging researches that are focused on developing clinical solutions to some problems? Would it work in a situation whereby you develop the phage, and if pharmaceutical companies abroad can use GMP methods to formulate it, and you come and try it down here? So I think that may be a method to work, because going by following the complications following phage research in clinical practice, it may not be difficult to discourage. It may be discouraging for people to go into clinical, finding clinical solutions to problems. So, don't you think partnering with companies, pharmaceutical companies, for them to get your phages in GMP conditions that can be used for clinical practice may be an option to go? Thank you.
Good point, Emmanuel, and I'm glad you clarified that, because yes, I do not want to discourage people from working on phages that would be for direct human application. The reality is, though, that whether those phages are manufactured at GMP in the US or in Africa, it's about 10-fold more expensive to manufacture by GMP than non-GMP methods, or wherever in the world you manufacture it. So there's just an added cost there. There's a caveat to that answer, though, and that is the new regulatory system that has been put in place in Belgium for phage manufacturing. And it's more for a personalized individual approach to phages, where GMP manufacturing is not required to develop phages for an individual patient. I don't think individual phage manufacturing for patients in Africa will be realistic, but it might be possible to apply that Belgian system on more of a regional level. So Jesca and I and others have talked about having phage banks. Jesca mentioned that in her talk, that could be rapidly deployed if there's an outbreak in a region, rather than for individuals, such as is being done in Belgium. So I do think that's a path forward, possibly, for phage applications, especially in people in Africa. Essentially, it's similar to what has been done in the former Soviet Union for many years, is to have phage banks that were then made available under non-GMP conditions. And it's a whole hot topic to discuss GMP or not for humans. The cost, no matter what, the cost is 10-fold higher, which, given that grants are hard to get, will always make that a bit of a challenge. I hope that answers some of the questions.
Just to add that for compassionate use, I think it might be a bit faster to be able to test your potential phages in a situation where antibiotics really won't work or can't work for the particular strain in question, or the bacteria or infection in question. So, but before you get there, you need to process your phage to a certain standard and test it to a certain standard to be able to ascertain the possibility of this phage working, and also the potential benefits you will get from the use of this phage. So like in 2017, this work done here in the UK on the 50-year-old girl having infection that could not be treated with antibiotics. So you have a situation where this group of people have got these phages, they've tested, they've got some data on these phages, isolated the strain causing the infection, and then tested it and then applied it. And also the infection got resolved in the end. And I think Ben as well has done so many of these compassionate uses. So you can go through the compassionate use route, but in terms of clinical trials and drug development, you really need to do quite a great deal of work to be able to reach to that point. So we've published a paper just about 2 weeks ago on preclinical data and safety assessment of phage therapy in humans. So you might want to have a look at that and see all the procedures and data you need to be able to process your phage for clinical application. Thank you.
Thanks, Janet. And if Rob is still online, maybe he can tell us the challenges they had in Belgium, because everything has a bit of, we can talk of advantages and disadvantages, but maybe Rob could tell us how Belgium have succeeded through phage therapy.
Well, it's definitely a slow process. You have to realize we defined, obviously led by Dr. Jean-Paul Pirnet, the first phage cocktail that was used in 2009. But yeah, after a few compassionate use cases, this has gradually scaled up. They have obviously the advantage of having specifically designed clean rooms that have been developed for tissue culturing and that are being used for GMP-like manufacturing of specific phages that are made available. But that number is small. Also, the magistral preparation law that Tobi was referring to, actually, it's not a new law. Magistral preparations, they exist and they contain active pharmaceutical ingredients. The only thing we did was gotten phages approved to be introduced in, so it's not a new law, it's just implementation of an existing law to include phages in these products. But they are also, as Tobi said, individual patient products. So yeah, Jean-Paul will say that there are now about 80 patients that have been treated over the years, and this is ramping up as a cause of this. But it's still incredibly restricted in terms of production and the number of phages restricted to maybe 1 or 2 staph phages, 3 or 4 Pseudomonas phages, 1 or 2 Acinetobacter phages. So it's not warp speed, I'm far from it. That's
okay.
it.
Yeah, and I have another question on the nanopore. Somebody said they have, Martin said they have isolated phages and done sequencing via nanopore platform. However, he has challenges in that the phage maps appear to be half. Some genes appear to be missing. What is the problem, or where is the problem?
That I cannot say without seeing the specific data, to be honest.
All right.
Yeah, sorry to disappoint.
Yeah.
But
and
if
maybe
he sends
yeah.
me an email, I mean, I'm happy to discuss this.
All right. So Martin, kindly send Rob Lavigne an email. What situations could have accelerated phage therapy in India? And can we learn from their situation? It's an open question. Jessica, Tobi, Rob, whoever. Tobi?
I don't know a huge amount of what's been going on in India, but my understanding is that a lot of it is focused on compassionate use. So again, individual patients rather than larger clinical trials with a formulated or a preset product. But they've made huge progress. I think they've treated at least 50 patients maybe through, maybe somebody can help me. I'm forgetting the name of the organization there that's helping to make that happen. Vitalis phage therapy. So again, it's a personalized approach, I think, is largely what they're taking in India.
All right. And we've listened to Jessica's presentation, but however, she has, in an African context, we realize that we have some challenges. And now, because we have a few people on board who are not in Africa, we are wondering, how is it possible, like if it's possible, like to just have maybe someone to give us, like, can sequence maybe 1 or 2 phages just to help, because sequencing and TEMs are very hard in Kenya or in Africa or in Uganda, because apparently all journals say you have to have this, you have to have this, you have to have this. But because now we have you on board, we are wondering how you'll be able, maybe a training workshop, or having maybe just 1 or 2 phages being sent. Maybe there's a concern, maybe a question raised from the group, if it's a way possible that if there's an interested partner, whether it's in plants or livestock or humans, maybe you can help us get to them for us to be able to get to the other side of phage therapy.
Yeah, I think we're happy to make those connections. And as you suggested, Angela, it's probably best if the African researcher searches through the literature and finds someone who is working on a similar type of problem, phage problem, that they're working on. They can either try to contact those people directly, or they can go through us, and we can try to make an introduction to help smooth the way to ask if they would be willing to help with some sequencing.
Okay, thank you. I see Betty Kutter's hand is up, and thank you so much for joining this meeting. And it means a lot for all of us, just for you to be able to be part of us.
It has been wonderful. I happened onto it right at the beginning, and I hadn't figured out how to get in to say anything, but I am so thrilled and excited with everything I'm hearing and all of you are doing, and particularly Angela, seeing the leadership role you've been taking all of this time. It's, it's amazing. And then the people who are still, in a way, like Janet and so forth, to be very actively involved, and Tobi, to be actively involved in helping you. We are going to have we're planning to have an evergreen meeting this summer that will be held largely online, and it will be August 2nd through 5th. And we're going to be charging very little for it and nothing for people, but assuming we can manage to get it mainly broadcast and online with only about maybe 75 or 80 local participants, I'm hoping that we can make it very generally available. And that it would be very good for those of you who are far away to hear, to send things that you would like to have posted. We're talking about doing posters online. Could people hear me?
Yes. Thanks, Betty, for reassuring that there's going to be an evergreen. Thanks for that reassurance.
And best of luck to all of you. It's wonderful to be in the middle, and rather almost embarrassing to hear at the beginning talking about how the role I played. It is very, now at 81 years old, it's very nice to hear that.
Yes, and we are like completely, like, I don't know if we could send you gifts or lions and elephants, like they would be probably all the way into the US right now, because we really appreciate the hard time that, and also the time that you took off from your work just to train people and educate people on phage therapy. If there was a present, maybe it would be like 100 elephants or.
Well, for me, the biggest present for now has been listening to all of you, and every now and then hearing my name come up and realizing that from my small university, I have managed to make a difference in the world in this kind of thing.
And we are completely indebted to you and to Tobi and to everyone who wants to support the African Phage Forum and Africa research in general.
It's amazing. So anyway, I will keep figuring out ways once we figure out how the meeting will be held in August 2nd through 5th, and to invite you all to freely take part for no payment.
All right. Thank you so much.
This is Tobi again. I do just want to add my kudos to all of you for getting this Africa Phage Forum. Jesca, great talk. I think Emmanuel, Rita, and Julia and Angela have been involved with leading this, so, and others, I'm sure. So congratulations, everybody. This is a great step forward.
Yes.
But however, I would just like to urge, if there's any European, Asian, American, South American supporter who would want to work with low and middle income countries, I think we need to just set that portfolio, because we can do the research, but we always need this one partner from other countries who could push our budgets higher than what we're applying for. And maybe that's what Jessica said, like she had not really foreseen, like, because now she can't have a master's student because of budgetary constraints and everything. But I think I work with a project with IDRC, but because of working with another partner, then we can see you can already put your budget to adjust it towards having one PhD person, 2 masters, 3 bachelors, and for capacity development, I think that is really helpful.
I don't have the access to be able to put actual money of any significant level, but I would be very glad to have my name and some writing and supportive things built into any of your grants where it would be useful.
Oh, thank you, Betty.
I'm also going to add, I don't know if Rob is still on the line, but regarding applying for small grants or large grants, I actually do think that going for those small grants often is an easier first step, especially for younger participants or scientists. And I think, as Angela can attest, having international partners, and Jessica too, makes it easier to get those larger grants. And so, yeah, there's pros and cons to both large and small grants.
Hi, Jessica.
Which one? Jesca Nakavuma or Jesca Sasha? Betty, how
Maybe
are you?
both of you.
Betty, how are you?
Yeah, maybe both of you.
It's been so funny hearing another Jessica, and I'm like, no, they're not talking to me.
I was talking to Jesca Nakavuma.
Oh, thank you, Betty. I'm fine, and you?
I am doing quite well. I got my vaccination with Johnson & Johnson on March 13th, and my lab has started reopening. You know, it started in 1973, but it was closed from last spring through the fall. And I have just a couple of students working on it, but we will be we are beginning to get some work done, and I am healthy and excited. I take about 5-mile walks several times a week. I'm in good health. Yes, and I have wonderful memories of all of you.
Okay, thank you. We too. We also have good memories.
Yes. I think because of there's a curfew going on around in Kenya and everywhere else, I think I'm so grateful for everybody's insight. And Jessica, your presentation was wonderful on the challenges and what the advice that you gave us in terms of what we need to do and what you didn't think that was important and now is really important in terms of grant acquisition and everything. And I'm hoping that through this meeting, because we are going to have a network, everybody, if anyone has any problems or would like emails for any person who spoke today, you can kindly approach us, and I'm going to give you, I can share their emails with you. And yeah, and this is where, for one, I would end the meeting because I'm really like stuck in the dark somewhere trying to get home because of the curfew.
Okay.
Yeah,
Yeah,
Jesca
Yes.
Nakavuma, do keep in touch with me. Do write to me on internet. Okay?
Yes, I will. I will. I'm rare on Skype, communicate over Skype.
And Angela as well.
Thank you. And much appreciation. And Rita, do you have any closing remarks that you would like to close up and we end the meeting?
Oh, yes, please. Thank you so much. Thank you so much, Angela, for tackling the technical aspect of this talk. But most importantly, I want to thank so much Dr. Jesca Nakavuma, who has brought to light what it means to conduct phage research in Africa. And I also want to extend my thanks to Dr. Tobi. There was Rob. I haven't met you. And
Betty.
who else?
Betty.
Yes, and also, yes, Professor Betty. Yes, there was Janet. I really want to thank you so much for all your contributions to us. I mean, Africa, phage research in Africa is still young. It's still, it's a novel, it's a virgin area. It's still very young. And I must say that most of us fall in the early career. So coming up together was something that we knew would help us lift us up from where we are and probably get us to a level where we can, start. And so I want to take this opportunity to thank you all, but to also thank all the participants who have joined us from everywhere in the world to come and be part of this webinar. We continue to ask you to look out for our next series. Every month, we hope to have a talk. So we'll be approaching most of you on different topics as they come in. What we normally do, we look through your questions and then we think of something that is pertinent to us. So I want to thank APF. I mean, yes, that's African Phage Forum. That is Emma, Emmanuel, Angela, there's Notin, there is Julia. I want to thank you so much for all putting this together. And we also want to extend a very big thanks to the Phage Directory, and that is Jesca Nakavuma. So with all this, I think it is time to wish you a very wonderful day or a wonderful night, wherever you are, and to
And a wonderful
yes.
time to you. And let me remind you that for all these last 50 years of doing work with phage, I have been working in a small college where I am the only basic research lab and was working on relatively low budgets. And there's so much one can do with phage with enthusiastic undergraduates, and they learn so much.
Wow, that is very inspiring. That is very good to hear, because when we think of research, we always think we have to go big. Yet, from your expertise, you tell us that you can start small, and with the small can be amplified, to get students around and do the implementation and, get you to a point where you have so much to publish.
Yeah, and
So,
we have had phage meetings here for many years and so forth, but literally it has been, Evergreen has only a few thousand students, and as I say, mine is the only basic research lab there.
wow. So for the students out there, please don't ignore the small grants, just like you've had from the Phage Guru. I get to hear that, Betty, you're the originator or I've had very good things, but you're the one who literally started the phage, should I say the phage in the world, but something along that line. And we're really so grateful. And you can see your impact, how far the extent of your impact in the world.
It has
So
been
many.
very exciting. Yes, I worked with phage as an undergraduate, as a graduate student, starting in the early 1960s, 1963, and with phage therapy then starting in 1990. But it's something one can do with so little equipment and so little serious training and so forth, and it can go so far.
Yeah, and we really, really want to thank you so much for that information. I'm sure most of us now, we are ready to go ahead and look for all these grants, whether small or big, knowing that even small can do an impact.
Small is enough for much phage work, just because they are so simple to grow.
That's true. That's absolutely true.
And then if you have somebody who can occasionally take somewhere else who can occasionally take an electron micrograph for you or do a sequencing for you or things like that that you can't do yourself, it's easy to send phage and get those things done from people in the phage community who have big places.
That's so correct. And that's why we had to come up with an Africa Phage Forum, where we had to not only just think of the Africans, but everyone else whom we could talk to, collaborate to, write to. And I think this presentation has showed us so much that we can do. We can papers, and then from the papers get to know who to write to, and then go through people like Tobi to help with the introductions to, I guess, the Western world. So I think personally, I always benefit a lot from these webinars.
And I think it's very important for you to go ahead and isolate all sorts of phages in your parts of the world that are
Yes.
against your specific pathogens, and then get help in figuring out what they are and what their sequence is and how you can use them and things like that from outside. But to not so much get phages from the outside world, but do the very simple thing of isolating phages that I do with my classes of sophomores from the local rivers and lakes and things like that.
Absolutely. Wow. Okay, unless anyone has to say something, if there is anyone who wants to say something, I would want to wish everyone a good day, a good night, and a good time.
Good morning.
And good morning.
It's 10 in the morning here.
Yes. So Jessica, Sacha, I probably now would hand, this time over to you.
Yep, thank you. Yeah, I'll close down the room. What a great discussion. It's been awesome to hear it. And I'm so glad everybody could come. So let's keep doing more of these. Happy to help support.